Good afternoon, everyone. Welcome to the second lecture in this online lecture series on different aspects of life sciences. This is as part of the Azadi Ka Amrit Mahotsav that we are observing this year. Today we have among us yet another very eminent speaker, Professor Punyasloke Bhadury. Professor Bhadury would be speaking on microbial complexity in coastal oceans. What have we learned from mangroves? Now, about Professor Bhadury. He has completed his MSc from Edinburgh and PhD from Plymouth in United Kingdom, and subsequently he did his postdoctoral research from Princeton University, USA. He joined as assistant professor in IISER Kolkata in 2009, and since 2018 he is a professor of the biological science department in IISER Kolkata. He has also established and he leads the Center for Climate and Environmental Studies, an interdisciplinary center on earth and environmental sciences with societal relevance at IISER, Kolkata. He is the recipient of the prestigious Swarna Jayanti Fellowship Award in the area of earth and environmental sciences conferred by Government of India in 2019. He has published more than 800 peer-reviewed papers in international journals. His research interests include biocomplexity of microbial systems, biogeochemical cycling, and sea level rise, nature based solutions to ecosystem restoration, antimicrobial therapeutics, and developing technologies for sustainable groundwater. One of his technological developments have been commercialized for decontamination of human fecal matter in groundwater, STPs, and adopted by several government agencies, including the government of Nagaland. He's the editor of a series of very prestigious journals, as you see here. That's just the top of the iceberg, if you might say. It is a CV which is extensive. There are endless such very distinguished recognitions that you have for him. As you can see from the look of Professor Bhadury, he has achieved all this at a very young age. He's also the team leader of India's Arctic expedition, the winter phase of 2010. Welcome, Professor Bhadury. It's always been a pleasure to have you on the different for the different online events that we have had in our department. We look forward to having you for offline events as well. Hopefully that would come soon with the COVID situation normalizing. So without wasting any more time, without standing between the talk which my students and faculty members are waiting for, I hand over the stage to you with a warm welcome on behalf of the Department of Microbiology, Tripura University. Over to you,
Th-
sir.
Thank, thank you so much, Professor Chaudhuri. It's a pleasure to interact today with colleagues, young researchers, young minds in the Department of Microbiology in Tripura University, and also it is part of the commemoration of Azadi Ka Amrit Mahotsav 75th year. So I congratulate you and everybody in the department and in the university for organizing this online seminar series that will, I am hopeful, will inculcate young minds to take up research in microbiology, in particular in our area such as environmental microbiology. So without any more delay, I'll get started. Give me just a minute. I hope you can see the slide. Uh, is it visible? I hope it is visible. So I'll get started. Uh, I have put this title in a way where there are several keywords there: microbial complexity, coastal ocean, mangroves, and I've put a question mark. So it tells you probably in the beginning of the talk how complex it is to understand microbial systems, and in an interface where the land and the ocean meets, it becomes far more complicated. I draw your attention to the coastal ocean. It's really soothing to look at the coastal ocean. It's really soothing to walk along the sea beach, appreciate the beautiful weather. But when we walk through the sea beach or walk along the pathways of the coastal ocean, there's a lot more dynamic things that keeps on happening, which we may not be able to see, but we see it in a form of a macroscopic scale. For example, all the processes that goes on in the coastal ocean are mediated by diverse organismal communities, and because of these processes, we experience the monsoonal patterns, the rainfall, the agricultural productivity, the GDP, everything that happens in the country. And of course, the when the ocean system starts to change, and it has become so in the era of the Anthropocene, then many of these processes are altering and therefore having cascading effect on the climate and monsoonal patterns. Thus, it is important to understand the coastal ocean more intrinsically from the perspective of microbial complexity. So, how do we define coastal ocean? Well, essentially, this is about 7% of the total ocean area, but it's very, very productive. It supports about 10 to 30% of the total primary production, and it is supplying more than 90% of the world's fish yield. So, there's a significant contribution of the coastal ocean in GDP of the country, and also, of course, the socioeconomics and livelihood. But there is another bigger aspect of the coastal ocean. Actually, it plays a very important role in modulating the CO2 exchange that is happening between the ocean and the atmosphere. And intrinsically, it helps in cycling of carbon, which is very, very important because it is the change in the greenhouse gases, particularly the carbon forms, that is affecting the global climate. And we saw the recent IPCC report that draws a very dark picture of what is yet to come until and unless we change our course. So we therefore need to understand the coastal ocean more intrinsically and in a more broadened fashion, understand what kind of interaction that happens in the coastal ocean between biological communities and what are the functions of specialized organismal groups. Now, when you look at the organismal abundance in coastal water, it is quite mind boggling. So, if you look at it out here, bacterial cell abundance in coastal water can vary between 10 to the power 5 to 10 to the power 6 per ml. I think it goes up beyond that in certain coastal oceans. In sediment, actually, the number is even far more higher. So, obviously, sediment has lot more nutrients, and therefore that drives the productivity. But there is a lot of interaction that is happening between the sediment and the water, which we call it as the benthic pelagic coupling of the microbial systems. If we look at other organismal forms, for example, photosynthetic organisms, of course, the photoautotrophs, the microalgal cell abundance, it can be around 10 to the power 6 liter, but this number also can change depending upon which part of the coastal ocean we are looking at. And of course, on the based on the algal cell abundance, there is the plankton abundance, mostly the ciliates, which are dependent on the algal cells. Their number also varies. So essentially, in a liter of seawater or in a milliliter of seawater, you will have lot more cells that are present, and we really do not understand how the interaction that happens and how the ocean sustains such rich biodiversity, and what is the scale of the complexity of the organisms that reside in them. Of course, it is important to understand the coastal biodiversity because it is the coastal biodiversity that intrinsically, or rather I should say directly, influences our human health. And we have seen how when the nature changes, how the human health can be affected. It is nothing new. Last 200 years we have seen the changes. So all the outcomes are directly dependent on the biodiversity, and coastal ocean plays a very important role in sustaining the biodiversity. And of course, the bigger picture is the livelihood component. Many people who live along the coastlines, particularly in India, for example, they are directly dependent on the coastal resources for their daily needs. So there's a big dependency on the coastal resources, and therefore understanding how we can maintain the good health of the coastal ocean is very, very important. And all of this contributes to the major 17 goals of the United Nations Sustainable Development Goals. Of course, here I want to draw your attention, goal number 2, zero hunger; goal number 3, good health and well-being; goal number 5 and 6, gender equality, clean water, sanitation; 13, climate action; 14, life below water; and of course 17, which is a partnership for the goals. But there is also another interesting aspect is there, which is mentioned here, is goal number 9 and goal number 11. Industry, innovation, infrastructure, and sustainable cities and communities have a very important role to play for goal number 14. So I think all these components are interlinked with each other, and coastal ocean actually directly or indirectly influences many of these goals that will transform our world. And I want to remind you that India has committed to these goals, which is to be achieved by 2030. So we have a big task ahead, and therefore you need to understand our coastal oceans more deeply. And of course, we need to come up with better sustainable approaches for utilization of the resources of the coastal ocean. Now, I want to draw your attention that generally, when you look at the biological communities in any system, there are many factors that control the structure and functioning of the biological communities. And of course, that is the case for coastal ocean. For example, freshwater flow is a very, very important factor. Let's say a coastal area which receives a lot of fresh water from the major rivers, that's going to be very dynamically influenced. There could be groundwater influences. There are a lot of aquifers are there, and from these aquifer, seepage of water happens in coastal areas, and that also influences the biological communities. As I said before, that the coastal ocean sits on the boundary between the land and the ocean system. Therefore, there's a lot of nutrient inputs that is happening from the land boundary. So, for example, the types of carbon, the types of nitrogen, that would have a lot of influence. Obviously, in the last 200 years, the anthropogenic forcing and the types of nutrients that are loading into the coastal ocean has increased dramatically. And we know in the last 10 years, one important aspect that has become to our picture is the plastic in the ocean that is creating a lot of menace. So I think we are looking at a huge anthropogenic pressure that is emanating on the coastal ocean, and therefore that is also influencing the structure and function of the biological communities. Besides that, pH is another important aspect, because of the so much emission of the greenhouse gases, and the ocean absorbing a lot of CO2, for example, that is also affecting the pH. And we are facing a situation called ocean acidification, and in the coastal water we call it as coastal ocean acidification. So that is something which will have huge implications, because we all know in a cellular system, pH is such an important factor that controls many of the processes, enzymatic activity, metabolic pathways. Besides that, there are some factors which are very, very typical for coastal ocean, very specific to coastal ocean. For example, the depth of the water, the tidal fluctuation, wind, current, all these things have direct or indirect consequences for the coastal ocean. Now, if we look at it from a broad perspective, from a functional perspective, of course, this is what is happening. A fantastic paper that came out in 2015 by Science by Alexander Warden's group. And here it very nicely shows the microbial loop and the carbon cycle. But we know that this loop actually works very nicely in the open ocean, but in coastal ocean, our understanding of the loop and how carbon cycling actually happens is not very clear. So, for example, we really do not know how bacterioplankton. So these are bacterial communities that are planktonic in nature. Their entire life cycle is planktonic, and they are present in very high abundance. What is their structure? What is their function? What they are doing? How they are contributing to the carbon cycling? How they are contributing to nitrogen cycling? How they are controlling the other components of the loop? For example, the DOM pool, dissolved organic matter pool, the fecal pellet breakdown, the interaction between the bacterioplankton and the higher group of organisms, symbiosis. All these aspects are not very well studied, because if we do not understand these processes, we'll not be able to understand the greater picture, the interaction between ocean, atmosphere, hydrosphere, and the land. And that is something which is the earth system processes. Now, When we talk about complexity of organisms in coastal ocean, we say it from the perspective of biological complexity, and essentially it is the functional relationship between complexity in biological systems, for example microbes, and the interaction and the processes that those control. It seems likely that these biological communities that we find in the coastal ocean, they of course will be related to the physical and the chemical complexity of coastal ocean. So for example, there's a strong correlation between the cell size and the elemental stoichiometry, nutrient stoichiometry, and that in turn affects the ecological stoichiometry of the coastal ocean. Therefore, we need to understand these biological communities. For example, from the perspective of gills, microbial gills, what kind, how many types of gills are there? What, how unique are the gills in specific coastal biotopes? For example, in the coastal ocean, we can see estuaries, we can see mangroves, we can see intertidal areas, salt marsh, seagrass beds. So how the biological communities actually are driven in those kind of system, and what is the manifestation of such complexity? Do we see plasticity, how plasticity is being driven by physical and chemical attributes? So all these kind of questions are very, very important to address and understand. And in today's lecture, I'm going to spend some time on one of such questions, and hopefully able to give you some picture to you. Now, to be able to address such critical question, we need to look at the biocomplexity of communities from a very different perspective. We have to use time series, because time series are nothing but essentially long-term regular observations of the physical, chemical, and biological processes in coastal ocean. And with such extensive temporal and spatial data sets, we can then understand how environmental drivers shape complexity of biological communities. And obviously, when you start to address such kind of questions, then we can address very complex questions like carbon cycling, nitrogen cycling, or very finer component of the nutrient cycling, such as iron cycling, or what are the implications of this cycling on the fisheries production, what are the implications of this cycling on the coastal health. All those questions can be addressed. Obviously, there are many time series sites are there globally, the most famous being the Pacific Ocean has 8 time series. The Lobo is the very famous one. Atlantic has the CPR, it has got 16 time series. European seas has 18 time series. Indian Ocean has only 1 time series, and that's an open ocean. The coastal ocean time series particularly focused on mangroves. That I believe there is only 1 time series is there, and that is the Sundarbans Biological Observatory time series, which was started by us way back in 2010, and it has completed more than 10 years now. So that's probably the only time series focused on mangroves, and probably in South Asia also. So we work in the Sundarbans. Many of you have heard about Sundarbans. It is the world's largest contiguous mangrove ecosystem. It is a UNESCO World Heritage Site. It is a Ramsar site, and of course, this mangrove is very, very dynamic, very young. It appeared in the late Holocene, less than 7,000 years old, and it sits on the delta of 3 major rivers: the Ganga, Brahmaputra, and the Meghna. And these rivers are pumping about 140,000 cubic meter of per second of fresh water. So you can imagine the amount of fresh water, probably second or third highest in the world. And this is um, something that makes the Sundarbans very, very unique. It's spread across 10,000 square kilometers. Of course, most of it is in Bangladesh, but quite a bit is there in the Indian Sundarbans. And here, the part which is marked as yellow and green and orange, these are considered part of the national park of Sundarbans. We call it the Sundarban Biosphere Reserves. And of course, when you talk about the Sundarban, the first thing comes to our mind is the swamp tiger, or the Bengal tiger that is found in the Sundarbans. They're very, very unique. They have adapted themselves to this unique ecosystem, and there are more than 99 islands are there in Sundarbans, and the tigers can move from one island to another island. So that's something which is very, very unique in this system. And in the Sundarbans, in the western part of the Sundarbans, the largest island is the Sagar Island, and down south of the Sagar Island, we have set up this time series site, the Sundarbans Biological Observatory time series. So when you look at the Sundarbans, any image or photo, this is what we see. The mangroves are immersed in water. Of course, this is the saline water that is there. The name Sundarban is coming from mangrove Sundari. Okay, and they're very very unique because they are adapted to fresh water and also to saline condition. And last 50 years, the salinity has been changing in Sundarbans, of course, throughout the Indian Ocean, and that is also affecting the mangrove vegetation and particularly the Sundari vegetation. So here, we interlink the mangrove with the tiger, as I said before. But what is more important to understand as a microbiologist or as a practitioner of biology trying to understand microbial system is that in the Sundarban, this microbial loop, how it is working. One aspect that is very very important is the mangroves actually shed their leaves several times in a year. The reason being is they are taking up the water which is saline water, and the salts are accumulated, of course, in the leaves, and the leaves needs to be shed, otherwise it will become very very toxic. So what is the fate of these leaves? Because these leaves are rich in carbon and nitrogen pool, and we really do not know how biological communities, in particular bacterioplankton, the planktonic bacterial communities, how what is their role in the breakdown of the litter fall. Although there is lot of suspended particulate matter in this water, very very high suspended particulate matter ranging between 200 to 700 milligram per liter, probably the second highest in the world. And how the litter fall is broken down by bacterioplankton, because that will release the complex forms of carbon and nitrogen. But there is one caveat is there: these litters are rich in tannin, so that can also inhibit activity of biological systems, particularly bacterial systems. So this aspect of the loop is not very well understood, and this is something we have been studying for the last 10 years, trying to understand it. Now, here, as I said to you, down south or down west and down south of the Sagar Island, we have set up this time series inside the Sundarbans Biological Observatory time series. There are three stations are there. These stations are strongly influenced by the freshwater from the river Muriganga, and there are tides happening twice daily. So saline water is entering from the Bay of Bengal. There are some planted mangroves are there, and there is also human interference is there. So we selected a site where you can study all these factors, and you can understand how the biological responses are, how the physical chemical attributes changes, and of course the atmospheric attributes in this system. So this has been set up way back in 2010, and we have been studying this system for quite some time now. This is a typical profile of the salinity that you will see in the Sundarbans. What happens is during the summertime the salinity is highest, of course. There's less water flow is there, fresh water flow is there, lot more evaporation rate, but also that time thermocline develops, so salinity can also become zero in the upper layer of the water. But monsoon is the time when there is incessant rainfall, and that brings the salinity down to zero, but the depth of the water increases dramatically, and also the suspended particulate matter load goes down. So that time the salinity is much lower, and in the post monsoon the salinity starts to increase. And during that time of the year also, a lot of nutrient rich water from the Bay of Bengal reaches the coastal ocean or surrounding the Sundarban, so that drives the productivity. So this is a typical profile that you get if you study time series, or if you do timely measurements. You can draw or you can get the snapshots of what is happening in the ecosystem. Now in our lab, we are trying to understand the biological complexity from the perspective of carbon cycling and litter fall breakdown. And the methodologies that we adopt are very very straightforward. What we do is we go to our site, we collect a lot of water, we actually filter the biomass, and then we do lot of these downstream molecular tools, use molecular tools. So we do PCR and then sequencing. Of course, clone library is a very traditional method that we still continue to use in our lab, that is based on Sanger sequencing. Last since 2014, I would say, we have switched on. Initially, we switched on to Roche 454 sequencing way back in 2010. Then we switched to 2014 onwards. We have switched on to the Illumina sequencing, and now we also do nanopore sequencing. So we use all the three platforms for sequencing. Obviously the techniques are all very different, but generally we start from environmental DNA and then we go to the community structure analysis. We use a lot of computational biology approaches when you're trying to look at the structure and function of communities. At the other end, when you're collecting water from our time series sites, we also measure a lot of critical environmental parameters that is helpful to understand the relationship between organism and their interaction with the environment. And of course we measure a lot of the environmental parameters. We use isotope ratio mass spectrometry to detect the signatures of the carbon isotopes. We use a lot of the TOC and the total carbon measurements to understand the carbon profiles. Of course, spectrophotometry is something that we continue to do, and we have also been doing since 2012 inductively coupled plasma mass spectrometry, and of course optical emission spectroscopy to understand the profile of particular forms of metal species such as that of iron and copper, because these are very critical components of the metalloenzymes present in microbial systems. Now, I start giving you a traditional output or a traditional approach. So this is a clone library approach. We have sequenced 400, 550, 45 clones. We have got sequenced 40 clones from each library. So, and we made about 14 libraries. And it's a very straightforward approach. If you sequence them, if you do the relative abundance plotting, it is not to the surprise. It's a proteobacteria dominated system. Any place we'll go, we'll see that proteobacteria dominates. The reason being is their metabolic flexibility and of course their adaptability, the cell shape and surface area. These are very critical factors, and very efficient ABC transporter systems that are there. So we got an understanding that in this system it's proteobacteria system, but there are a lot of other communities are there which may be present in very low abundance, might be very important, but essentially the system is largely driven by proteobacteria, but also there are signals of firmicutes which are very important in any coastal ocean system. When we look at the proteobacteria from the perspective of class and of course other groups, we see that this system has dominance of gamma proteobacteria compared to alpha proteobacteria. This is just the reverse what will you find in any coastal ocean globally. So it's something very unique for Sundarban, and we think that is because of the huge freshwater flow and the monsoonal influences that affect this system. And we see that certain times of the year the system completely transforms from being very diverse to only one type of community. And this is one nice example from 2012-13. The study result shows that how the transformation takes place, and this continues to happen. So this entire result is largely based on, as I said, Sanger sequencing. Now what we wanted to do is we wanted to check whether the information that we are getting from the Sanger sequencing, whether that is has any parity with robust sequencing techniques, new second generation sequencing such as Illumina, for example. So we also in parallel compared our data with Illumina sequencing. Of course, when we do the Illumina sequencing, the reads are much more. So we see, for example, in clone library, we can get about bacterial family, about 13 of them. But in case of Illumina MySeq platform sequencing, we get about 64 of them. So lot more depth. But the in the library, clone library, the families that we encountered, all those families were there in the Illumina. So it's not like clone library is missing out the broad groups. So it's not too bad. The traditional approach is not too bad. But of course, the deeper the sequencing is, the greater the picture you start to generate or to understand in this system. And you can understand that the abundances are more particularly for groups which are very important in carbon cycling. Now, so that the take home message of these two are proteobacteria dominated, gamma proteobacteria dominated system, just reverse. And certain times of the year it is only gamma proteobacteria in the system, and there are firmicutes are there. Of course, if we look at photoautotrophic organisms which are there, which are planktonic photoautotrophic, particularly cyanobacterial system, here we see that their numbers are much less. They are not very significant player except during the post monsoon time, and the system is dominated mostly by, among the photoautotrophs, dominated by the pico cyanobacterial members like Cyanococcus, and you see huge population diversity in this system. Now what we wanted to do is the information that we got from the Sundarbans, we wanted to compare against some global estuaries, mangroves. So in this case, what we have done is we have done data analysis. We have generated the data, analyzed it from Columbia estuary and the Delaware estuary in North America. We have selected Geelong estuary and Pearl estuary and Hangzhou estuary in China. Of course, Geelong and estuary has mangroves in it, and of course you have got the Sundarban estuary. One distinct thing that comes out from the Sundarban deep sequencing, and this is all Illumina data we have generated and processed, is that we we get the dominance of Vibrio out here, and there's a lot more Firmicutes are out here. And I know you must be wondering what this code stands for: MET, MAEHE. These are the four major estuaries that are there in the Sundarbans. So we have looked into all these estuaries that are also, of course, present in the 4,000 kilometer area of the Sundarbans. And one thing is very important to draw attention to, that here Vibrio dominated system, and there's a lot more Firmicutes are here compared to other mangroves that are globally, and of course other estuaries do not have much Firmicutes. So that is something which is very very important. So there's a typical biogeographic pattern that is developing, or that we see in case of the Sundarbans. And now, if we take this data and try to look at the perspective of, let's say, the geochemical signals, say nitrogen profiles, if we look at the time series side, we'll see that the nitrogen profile is largely kind of the similar the trend. Once in a while, it goes very very high, but largely it is less. But compared to other coastal ocean, the nitrogen is quite high, and that is because the Ganga is dumping so much of nitrogen, and much of this is anthropogenic nitrogen. So it's a nitrogen rich system, and that has also got some implications in the functioning and structure of microbial communities, particularly complexity of bacterioplankton. And here in this case, what we have done is we have done plenty of sequencing. We have selected several sites in Sundarbans and a site which is far away from the Sundarban, and we wanted to see what kind of groups are there. And we see that the system is dominated as usual by gamma proteobacteria. So actually, coastal Bay of Bengal has dominance of gamma proteobacteria. There are actinobacteria also are there, and there are members of Bacteroideae, Ignavi bacteria, and Vericomicrobia. These are very very important because these organisms are known to play important role in breakdown of complex carbon. And on the left hand side, what we have done is we have plotted the nitrogen profile based on high and low nitrogen concentration against different groups of bacteria. And we see some cases we see very positive correlation, and some cases we don't see any correlation. So nitrogen tend to, in many cases, affect the structure and function. For example, if you look at Spingobacterialis, or if you look at Vibrio, we see that there's a strong correlation that is developing also in case of desulfomonodylis. The too much nitrogen means some group of organisms will start to dominate, and of course they become very very important player in many cases in this system. So there are specific functional complexity that has been developing in this system, and that is driven by the anthropogenic nitrogen in the system. What we did is we looked at this data and tried to generate a metabolic profiling. We wanted to see what kind of enzymes we can detect that can be linked directly to nitrogen functioning. And we see that in many cases, high nitrogen or low nitrogen concentration has strong correlationship with enzymes such as nitrate reductase or with the transporter system. So there is a strong influence of low and high dissolved inorganic nitrogen in this ecosystem. Now, once we have known that, then the question that we wanted to ask, the fundamental question which led us our study is, out of all these communities, which are the major players, which are the ones which are breaking down the litter fall? Because you've got so much mangrove vegetation and so much litter fall that is accumulating, that must be cycled, that must be broken down. And who are the players that are broken down? Because if we can understand, then we can start to estimate the rates and fluxes of carbon and nitrogen in this system. So we started to look at litter fall, and of course that when you look at any leaf or any even in mangrove leaves, actually tannin is a very very important component of the litter fall, and it is a source of carbon. So if it is broken down, those carbon can be taken up by the residential bacterioplankton communities, and of course in the process these complex carbon are being broken down, and they are also contributing to the DOC pool, the dissolved organic carbon pool. But there is also this recalcitrant pool is there, and that is something which is very important to understand. So what we did is we did some lab mesocosm experiment. So from Sundarbans, we transferred about several hundred liters of water down to our campus in IISER Kolkata, which is almost 160 kilometers away. and we set up this controlled experiments, and we ran it for 2 to 3 weeks. And the idea is you have got the control setup, and in the enrichment we actually add the tannin into it. So the natural water will already have some tannin, but into that we add some tannin, and we have estimated the tannin concentration based on the profile of the tannin throughout the year in our time series site. So typically once you've added the tannin, we then start to look at the profiles of gallic and tannic acid. So these are the breakdown profiles, and we see in course of time they are starting to break down based on the experiments that we undertook using the lab mesocosm approach. This is the profile of the nitrate in those lab mesocosm setup. You can see the nitrate is very high, but in course of time the nitrate is going down because there is a utilization of the same is happening. When we start to look at the biological communities at the Illumina scale sequencing, we see there's some interesting results are there. Of course, it's a proteobacteria dominated system, but we see this in many of the many days during the course of the experiments in our barrels, the lab mesocosm setup. There is dominance of proteobacteria, and there is an increase in the number of bacteroidetes. So gradually, with the course the experiment is increasing, the bacteroides numbers are also increasing, and that is very important because bacteroidetes represents a group, they can break down very complex forms of carbon. So we then started to kind of draw the relationship that they must be breaking down the tannin pool, because we see that the tannin is going down and the bacteroidetes number is going up. Here we also look at what is happening to the rare bacterioplankton in this case, for example. So we looked at that, and we see that there are some rare bacterioplankton are there, for example SAR 406, or for example there are some groups are there like fusobacteria, groups such as lentispheriaceae. These numbers, they're mostly kind of low in the system, but they can kind of increase in the course of the time. So gradually as the experiment progress, their number change. So the conclusion that we draw is that probably the bacteroidetes are able to break down the tannin, and they're making the carbon available in much simpler form, and then these carbon are being taken up, forms of carbon are being taken up by these organisms. So that is something which is very important. It starts to give us a picture of what is really happening in this ecosystem. We started to do some metabolic profiling, look at the functional genes, and we see that in day 9 and day 15, some of the enzymes, particularly the amino transferase, for example, and of course the 1,2 glucan synthetase, their abundance changes. Of course amino transfer is very important because that has huge implications in nitrogen metabolism, and glucan synthetase also directly or indirectly controls the carbon metabolism. So we can see that the genes are triggered on as the breakdown starts. So of course, now remember, this is a closed system. In a open system such as Sundarbans, mangroves, is going to be much more dynamic because there are a lot more other factors are there. But the conclusion that we draw is, of course, that in this kind of system, the breakdown rate is very slow, and you can see from the statistical analysis where you have done the NMDS using the dominant phyla, we see that there are some correlation between the gallic acid and the tannic acid to some extent. Of course, and we see that some of the communities, they are responding quite fast compared to the control, and they seem to be influenced strongly by the communities that are there with the addition of the tannic acid, because the breakdown is happening. So there is no doubt this process is happening, and it seems to us that this is a bacteroidetes mediated system, and that is very important because this group is very robust. They can withstand, they can break down different forms of complex carbon. So now we have some understanding of how the breakdown is happening and how the loop is working. Of course, we have a long way to go. I wanted to draw attention that when the breakdown was happening, we also looked at other components. These are the biological communities which are not necessarily bacteria, but phytoplankton, so microalgae, and the plankton which are eating the phytoplankton, the zooplankton. And we see that in course of time, actually the phytoplankton number kind of shifts in the course of time. If you look at the control, the number goes up. because tannin does not inhibit the phytoplankton growth, but when we add tannin in the water in the natural system, it also has some effect on the phytoplankton and also on the zooplankton. And I wanted to show out here, if you look at the stable isotope measurements we have done, we see that the delta 13C isotope values clearly indicate that this is a heterotrophic driven system. So the carbon signals are all coming from bacteria, and that has to be because of the tannin breakdown that is being undertaken by the bacteroidetes in the system. So we are now able to join the different components beyond bacteria and trying to understand how the complexity and how the interaction happens, so that we can then go back to the field and try to understand the bigger picture where the dynamicity is much more and huge. So I conclude out here, what have we learned so far from the mangroves? Well, one thing is very important, is that the mangroves are extremely dynamic. We It is very difficult to emulate, or copy the all the parameters in a lab condition, but if we can address some of the critical parameters, we can see that actually the physical and the chemical gradients of the coastal ocean, they have very important role to play. What I did not talk about in this interaction of today is how the plasticity is being driven. In this case, we have done a lot of genome sequencing of many of the culture isolates that we got from these experiments of tannin addition in the lab mesocosm, and we see there are specialized pathways are there that are very very unique, very very interesting. For example, aromatic hydrocarbon degrading pathway, of course not surprising, because there is anthropogenic forcing is there. There are superoxide dismutase which are originating, which are modulated by nickel. That's something which is very very unusual in coastal ocean. And we see that the drivers of environmental gradients, actually coastal ocean is being facilitate the organismal level association or the interactions, and the organismal groups exhibit functional complexity. It is very difficult to actually understand the complex interaction, but we can start to draw some initial pictures so that we can build the first set of pillars and then go on and do some complex studies and understand the interaction that is happening. So our study now has given some understanding. Now we have some data where we can now calculate the fluxes and the rates coming out from the tannin breakdown, and how that is contributing to the carbon cycling, the coastal ocean carbon cycling, and ultimately carbon cycling in global ocean. I want to draw attention that if we want to have a very deeper understanding of many of these processes, trying to understand the interaction between biological systems and the environmental and chemical systems, we have to go beyond lab. And here what we are doing is we are developing sensors and automation, and which are putting up in Sundarbans. We are getting real-time data to understand the health of the Sundarbans, and in the process, this data helps us to understand what the interaction between the physical and chemical gradients and the microbes are happening. And these are mostly based on wireless sensors approach. So we are developing our sensors ourselves and deploying in the Sundarbans, and it is giving very interesting pictures and resolution of the micro scale interaction that is happening between physical and chemical gradients with the biological communities, that will ultimately help us to understand what lies ahead or how we can deduce Sundarban, because this ecosystem is changing very dramatically. Sea level rise, coastal erosion, heating up of the oceans, and all of you have know more number of cyclones are now hitting the Bay of Bengal. So the changes are happening more dramatically in an anthropogenically climate driven system. If you want to know more about our Sundarbans Biological Observatory time series site, please visit this site. We will gather information what we do, what kind of what is we want to do. We are very welcome, most welcome to collaborate. We are open to new ideas. We are open to collaboration. We are open young researchers visiting our lab, doing internships. So feel free to contact us on that, and feel free to visit our time series site. You can contact us anytime on that. And I want to end up thanking Dr. Ray Chaudhuri and colleagues in the Department of Microbiology in Tripura University, all the young researchers and the students who are listening. I also take this opportunity to thank IISER Kolkata for the initial funding support, Swarna Jayanti Fellowship of Department of Science and Technology, Government of India, Ministry of Arts and Science, Government of India, WWF India, and Wildlife Trust of India. They are policy making organizations, but they also support our research which we integrate into policy and ultimately into livelihood. I am particularly thankful because all this research that I am presenting are the hard work of our lab members, the past and present members of integrative taxonomy and microbial ecology research group. And here you can see Dr. Anesha Ghosh, who did her PhD. She continues to do her a postdoc, Swarna Jayanti postdoctoral fellowship in our research group in the Center for Climate and Environmental Studies. Orka Prabha Mondal, who is doing PhD looking at the effect of sea level rise on biological communities. Chakresh Kumar, who is understanding how the Gangetic system is influencing the structure and functioning of biological communities in Sundarbans and beyond, mostly from the perspective of pollution. Yash, he's looking at the freshwater to estuarine gradient and understanding the interaction between biological communities and carbon cycling. Some of the work I have presented here is of Anesha and also of Dr. Tarkeshwar Singh. This is Gaurav. He's also doing a very interesting work, very different from what I presented. He's looking at how the human microbiome signatures affect diabetes too. And of course, one of his study sites is in Tripura. And the sensor development part that we are doing in our lab, it is mostly this work we are doing. There's a very young, talented chap is there. He's a BSMS student, and he's working with me and developing this sensor. So it's all the teamwork that we are putting forward and trying to understand some of the most critical questions that is important to coastal ocean, and of course very important to the SDG goals of India and globally. And I thank you all very much for listening, and I'll be very happy to take any question that you have now. Thank you very much.
Thank you, Professor Bhadury. I see that there aren't any questions. Thank you for giving our students a glimpse of what has been going on in the lab. And this is exactly what many of them would be very much interested in. I'm sure they would get an understanding of it, would read about your publications and get to know more, and perhaps feel free to get back to you. Now, as part of our lecture series, since it's a part of the course as well, what we do is we give them a question which they have to respond back to and submit the response in next half an hour. So the question for today is, give a list of the techniques that have been used for doing the work that has been presented by Professor Bhadury. So your time starts now and you have half an hour to respond. So with this, on behalf of my department, we again thank you, Professor Bhadury, for taking out time to address our students, our faculty member, and this is on YouTube Live, and we can already see that there are others outside Tripura University who have been watching. So thank you so much for being there whenever we needed you as a resource person. Thank you so much, sir.
Thank you very much. Thank you. Thanks a lot.